In this video, we will be talking about CRISPR and RNAi. We will be going over some background for the video, the basics of each method of gene therapy, how they do their job, how we choose between them, and which one I would choose. First, let's get into background. As you can see, I have some information on this slide, and I will be touching on each of the bullet points. We will be going over two methods of gene therapy, RNAi and CRISPR. RNAi stands for RNA interference, and CRISPR stands for Clustered Regulatory Interspaced Short Palindromic Repeats. In the hypothetical situation that we will be going over in this presentation, we will be trying to eliminate the production of a protein produced by what is called gene A. To understand which of these methods would be most effective and which one I would choose, let's look at the basics for each method. For the basics, let's start with CRISPR. CRISPR is a naturally occurring system that was used to protect cells from viruses. The CRISPR mechanism is composed of a Cas9 protein, which cuts DNA, and a guide RNA, which aligns the mechanism. Now, for RNAi, it is important to realize this is a mechanism and not a type of RNA. The two main types of RNA in RNAi are siRNA, which is small interference RNA, and microRNA. This censors gene expression in the cell and could potentially shut off the gene function. With the basics out of the way, let's go into more depth about how these mechanisms work. Again, let's start with CRISPR, which is composed of guide RNA and a Cas9 protein. The CRISPR mechanism has a specific guide RNA, which then locks onto a complementary DNA sequence. The Cas9 protein then cuts the DNA. The cell then repairs the DNA and allows the gene function to change and inhibits any further protein production. RNAi is also naturally occurring because the siRNA and the microRNA are naturally occurring in the body. When the double-stranded RNA is produced, an enzyme called dicer cuts the RNA, which is then picked up by RISC, RNA-induced silencing complex. The RISC then finds matching mRNA that complements the picked up RNA. The mRNA is then cut and the cell degrades it. This removes the message the RNA was carrying, preventing protein production from the gene. With the function explained, let's look at how to choose. Choosing between these two depends on what you want the outcome to be. CRISPR does gene knockout, which is when the gene you're trying to inhibit is knocked out of the genome. The CRISPR mechanism has less off-target effects, which makes it fairly easy to use. Finally, CRISPR targets DNA. However, in RNAi, mRNA is targeted. Along with this difference, RNAi has the effect of gene knockdown, which limits the effect of a gene, but rarely does it eliminate it completely. Also with RNAi, off-target effects are more likely to occur because the complementation of the RNA isn't strict and it can have this effect in other mRNA. Between these two methods, I would choose CRISPR because it is easy to use, it has less off-target effects, which allows for more precise cutting because of the Cas9 protein, which completely inhibits gene function. It's also been constantly improved and has been used more within this field. Thank you.